Yeah, every morning I come out here, I feel like I wake everybody up. Here you go, Nancy.
Thank you.
So this is bitterbrush, and it's very important food for mule deer in the winter. The seed collection is kind of the first step in the restoration process. As a seed collector, I get contracts from usually the BLM or Forest Service to collect native seed that's going to be used in restoration. A lot of the contracts are 3-year contracts because each species doesn't give seed each year. I might find the sites and get paid for the sites 1 year, then have to wait a couple years before I actually can get the seed. And I wanted to collect, say, on the Deschutes National Forest, I would have to go get a permit. It usually takes a minimum of 3 site visits before I make a collection. When I thought that we were getting close to the seed being mature, I can usually guess, knowing when it's the right time to harvest. I believe that's the art. It's not just science here. During my career in the sagebrush steppe, which has been about 50 years, I've seen the disappearance and the degradation of acres and acres, and there's not very many pristine native areas left. And it's important to capture that genetic material. We want to preserve it, and I think it's really important to try to protect what we have and to try to restore areas that we can.
So we're out here collecting bottlebrush squirrel tail with the Bureau of Land Management, the Forest Service, and Fish and Wildlife Service, and we're here about 10 miles outside of Reno, Nevada. So we're really interested in this site because these are the plants that have the traits and adaptations for surviving in a really hot and disturbed area. So the seeds that we collect here do really well in restoration sites, just because they're so accustomed to disturbance. When we are collecting and choosing sites where we want to collect seeds to ultimately produce and then send out into restoration sites, it's really important to do a few things. If you have a big team, you'll split up the landscape so you're not sort of on top of each other, kind of create quadrants to work in. You want to find populations that are really large, and that also helps them sustain the sort of stress of collection.
Practices for not harming the population is you don't want to take that much seed from your site, especially if it's a smaller site.
We'll never collect more than 20% of the available seed on a site.
But for plants that you can't necessarily count all of those seeds, like a bush, you might want to do every 5th plant.
So at a site like this, it's really important to make sure you're collecting exactly what you're looking for without inadvertently getting this nasty cheatgrass in our bags as well. This cheatgrass seed looks exactly like squirrel tail seed. And we've learned from working with our seed cleaners that it's a lot easier to not get the weed seed in at this stage.
I want to make sure that this is a quality collection that has the full range of diversity from that population, from that species.
This is prairie June grass. I'm supposed to collect 3 pounds of this. Some of the species they want me to collect on the grasslands, and some of the species they want me to collect at higher elevations.
It is so easy to only look at the biggest plants out there. They really catch your eye, or the most blue flowers, but you really do want to collect evenly and randomly to ensure that you're getting all of those genotypes. And then you also want to ensure that you're collecting from at least 50 plants to ensure that we're getting 95% of the alleles within the population.
So sometimes you can think, if you look around this area, there's a lot of Poa secunda, but a problem we have with our grasses in the Great Basin is they don't always fill their seed, and especially under drought conditions. And so you can be scouting something and monitoring a species phenology and then go actually to harvest the seed and do the cut test, and it's all blanks. You think you have seed and you don't.
What I am going to be demonstrating is called a cut test. And the reason why we do this is to make sure that the seed we are about to collect is going to be ripe and fully filled. And then we take our double-sided tape, we put it down on our clipboard, and then we just pull out some of those seeds from our seed heads. And then once we find a couple of seeds, we take a razor blade and we cut those seeds in half. And then from there, we look at the inside of the seed. So this seed is completely white and full on the inside of it. And then this one is empty and black on the inside, which is a non-viable seed.
They'll do a cut test to calculate how many ripe and healthy seeds there are. And once they get all of those numbers, they can kind of estimate there are 80,000 available seeds in this population. If I only want to collect 20% of that, will that still meet my goals?
I have made collections where there's nothing there in my early days, so I don't want to spend a whole bunch of time collecting and then the seed not be good.
There's a lot of different methods you can use to collect seeds. Like grasses, you'll always want to hand strip them.
The grasses and the forbs are so specific that you almost have to pick one individual flower at a time. For shrubs, we have a hopper, or I use a big plastic dish that I stick under there. Well, we got a bunch there. You want all the seed to fall into your either hopper or whatever you have. If I'm whacking in and I'm not getting any seed, then it's not ready. I only use a racket for the bitter brush. Sagebrush has a really tiny little seed in the fall. I like to strip the flower heads off. This is made out of a feed sack, actually. One of the things I like about it is that it stays open. And so I'm collecting and I have both hands. I don't have to keep one hand here. It's important for the bag to breathe. You don't want moisture forming in there for most of the species.
We use paper bags because the seeds don't stick to them very well, and then also sometimes when you collect there's moisture in the seeds, and if you let that sit in a plastic bag it will mold and then kill the seeds that you're collecting.
If you collect at one site and then drive to another site, you don't want to just leave those seeds in the car. Like you wouldn't leave your dog in the car. It gets too hot. So what a lot of folks will do is they'll put them either in a lunchbox, under the truck in the shade, or just in the bags and kind of weigh the bags down underneath the truck. But typically you do want to get things shipped to a cleaning facility as soon as possible.
I've been at a lot of sites where I've collected seeds and then 2 months later the site burned down and those plants are just like gone forever. But part of me feels like, well, I did something. It's really hopeful to have seeds in a bag from that site because you took a snapshot of what was there and you can hopefully reestablish it. We could never capture enough seed from these wild populations to physically take the seed and put it on a restoration site. So then we take the seed, put it in production, make it a big collection that still has the genetics that we're interested in, and then use it for restoration.